This is a Mutate DNA, day 1, and this video will show how to set up and start your overnight cultures of bacteria that are expressing MutY with some kind of difference, a difference in the MutY structure. We're going to begin by adding antibiotics to 80 milliliters of the LB media. So this is provided by the educational lab staff, and I have my checklist here. I need to add 80 microliters of kanamycin, 320 microliters of ampicillin, and 200 microliters of the tetracycline. And this will be something that you do with your TAs. The TAs will be doing the addition of the antibiotics, but I just wanted you to see that even the instructor uses the checklist approach to make sure that we don't miss anything. Mission critical. Kanamycin. Here it is. Get that ready. 80 microliters. Check that off. Now we need 320 microliters of the ampicillin. Ampicillin. And then added that, so we check it off. And then finally, 200 microliters of tetracycline. Could use either device, but I'm going to use the big one because it's a little faster. Tetracycline is dissolved in 95% ethanol, and it's light sensitive, so that's why it's in the foil. Good. And now it's important to mix everything. And now I'm going to be distributing that amongst these 5 tubes. I have an extra tube that you as a student that the students won't need. The extra tube is just to verify that my technique is good technique, and I don't have any cells that can grow. This is going to be my negative control, like no bacteria. And then the other tubes, they're labeled according to some culture ID. I'm going to use my initials, MPH 1, MPH 2, MPH 3, MPH 4, and MPH 5. So those are my 5 cultures. And I need to put 2 milliliters in each of those tubes. You could use dial in 1 milliliter and do it twice for each tube. But I'm in a rush, so I'm going to use a 10 milliliter pipette. And the red bulb. So just like we did with pipette liquids, I'm going to start by fitting the red bulb onto the 10 milliliter pipette. Take up all the media that I need, and then pipette 2, 4, 6. I'm just looking at the markings on the graduated cylinder, on the graduated pipette until I get all the way to 10. And now each of the tubes has 2 milliliters. My negative control for that one, I'll just demonstrate what it looks like if you're going to use the big pipetter. You got to dial in 1 milliliter. And then you've got to go back and forth 2 times to fill it. 1, 2, so much slower. But also, it also works. Okay, now we have the tubes ready, and we need to inoculate them with some cells. And the cells are going to come from the colonies. The colonies are in this FPD plate. So I get my data sheet out, and I'm going to record the things I need to know on the data sheet. The date. Today is September 7th. I'm in the instructor team, but you'll put your section number, my name, and then the variant is printed on the plate from where I'm getting the cells. So I'm going to write here FPD, and that has a symbolic meaning. It means that in this region of structure that we're calling the FSH loop, the residues, the amino acid residues, are phenylalanine, proline, and aspartate. And the culture ID, that's what I put on the tubes. MPH1, I'll skip a beat, MPH2, MPH3, MPH4, MPH5. And all of these cultures, all 5 of them, will be from the cells in one of these colonies. To get the cells into the cultures, we're going to need 100 microliters of pure water. So pure water. Here we're gonna dial in 100 microliters. So it's a small volume at the bottom of the tube. Now we need to pick up the colony. I'm going to dial in a smaller volume, get a fresh tip, and it really doesn't matter which one I pick. And this time I don't really need to get the whole colony. I just want to pick up the cells from the plate, verify that they're on the tip, and then I rinse the cells into the solution here. Just like we did for count cells. And so the cells are now sitting in this 100 microliters of solution. You can vortex the cells if you're worried about getting them all distributed, but pipetting up and down probably just did a fine job. And now we're going to use what we call the dirty tip method for moving some of these cells into the culture tubes. For this, I need the P20 device, and I'm going to dial in 10 microliters. This is the smallest pipetting device. I'm going to go to 10. And now we pipette up and down the 10 microliters. And then instead of taking the 10 microliters, we push the liquid out so that the tip is actually empty. But there are some cells sticking to the plastic, and then we eject the tip into the tube so that the cells get carried to the bottom of the tube. Let's do that again. We're going to do that 4 more times. Pipette up and down. Remove the liquid. Leave the liquid behind. Center your tip over the mouth of the tube and then eject. Empty. No liquid, just cells. Pipetting up and down gets some of the cells onto the plastic of the tip, and then we leave most of the cells behind. We want to start with just a few cells. And then for my negative control, which you don't need to do, the TA will do this for us. You just take a tip and don't touch it to anything and just eject the tip, and that'll be the control. Put the caps back on. And then the TA will collect these from your bench and move them into the incubator at exactly the same time so that everybody gets a fair start, and they will grow for 18 hours. And you'll get these cultures in the second day, and you'll follow the procedure to wash the cells, harvest the cells, wash the cells, dilute the cells, and put them onto the KAR, the plates that contain rifampicin, the KAR plates and the KA counting plates. Mutate DNA.